Okay, so welcome to this next video on the G-inhibitory pathway. Okay, so now what we're going to do is we're going to see the consequences of cyclic AMP levels going down in the neighborhood of our receptor. Okay, so basically attached to the membrane of the cell, what you have is a scaffold protein known as ACAP12. Okay, so this is ACAP12, and what that stands for is A kinase anchoring protein 12. Okay, and basically this is the protein which holds protein kinase A close to the adenylyl cyclases and close to the initial G protein coupled receptor. Okay, so I might just draw in the G protein coupled receptor here to get that message across. So here's the GPCR. Okay, right. So ACAP stands for A kinase anchoring protein. I will put that in. A kinase anchoring protein. It has a number of other names. It's also referred to as A kinase anchoring protein 250, which is because of its molecular weight. So its molecular weight is 250 kilodaltons, so that's why it can also be known as that. ACAP12, I presume, is the order in which it was discovered. There are many A kinase anchoring proteins, so I presume it was just the 12th one to be discovered. It also has a final name that you'll see occasionally, which is gravin. Okay, right, so attached to this, protein is the protein kinase A, and there are two types of protein kinase A in cells, type 1 protein kinase A and type 2 protein kinase A. Now type 2 protein kinase A is attached to these ACAP12s and is localized at the membrane, whereas type 1 protein kinase A are free in the cytoplasm. Okay, so here is our protein kinase A, and basically in the inactive state, protein kinase A consists of, is in these R2-C2 complexes, and this is something that's quite confusing about protein kinase A. There are two types, there are two subunits of protein kinase A. It's a big structure, and basically there is regulatory subunit of protein kinase A, of which this is the R2, the regulatory 2 subunit of protein kinase A. So this is the second regulatory subunit of protein kinase A. Regulatory subunit of protein kinase A. Okay? And now these two regulatory subunits have dimerized, basically. Regulatory subunits dimerize. So we've got two regulatory subunits here bound together. And they also bind with two catalytic subunits. So overall, you end up with two regulatory subunits of protein kinase A with two catalytic subunits of protein kinase A. Okay, so it forms this complex where you have two regulatory subunits and two catalytic subunits together, and that's known as the R2-C2 complex. So this whole complex is the R2-C2 complex. It has two regulatory subunits and it has two catalytic subunits together. And this whole complex is the R2-C2 complex of protein kinase A. Right, and the protein which attaches the R2-C2 complex to the ACAP12 is the regulatory subunit. So R2, the second regulatory subunit of protein kinase A, binds to ACAP12, and that's why this is a type 2 protein kinase A. So this is type 2 protein kinase A. Okay, so the catalytic subunit does not vary between protein kinase A and 1 and protein kinase A2. It's the regulatory subunit that differs, and that's what determines whether it's free or membrane bound. Okay, now what happens is to activate protein kinase A, you need to release these catalytic subunits, because whilst they're bound to their regulatory subunits, they're inactive. And these catalytic subunits are the portions that are actually going to add phosphate groups onto serine and threonine residues of proteins, basically. And to activate this protein kinase A, what you need to do is you need to bind cyclic AMP to these two cyclic AMP binding sites on each of the regulatory subunits. So you've overall got 4 cyclic AMP binding sites. And when you do bind cyclic AMP, what happens is the regulatory subunit changes conformation and releases the catalytic subunit. So let me show that. So if cyclic AMP did bind, what happens is that the regulatory subunit adopts a conformation which I will, for a cartoon, show like this. Okay, so now I should show cyclic AMP bound in those 4 cyclic AMP binding sites. And when you get that change in conformation, and I want to stress that the ACAP12 remains bound to these regulatory subunits, what now happens is that the regulatory subunits split off from the catalytic subunits. So the catalytic subunits go off, and they're now free and active, and they will add phosphate groups onto things. So these balls here, which I'll colour in blue, those represent cyclic AMP molecules. Okay, right. So if cyclic AMP goes down within the cell, it's going to mean that, these regulatory subunits don't have cyclic AMP bound to them. They remain with their protein kinase A catalytic subunits, and the catalytic subunits remain inactive, and you don't get phosphorylation of serine and threonine residues on nearby proteins. Okay, so that's overall the result of the G-inhibitory pathway, that the activity of these catalytic subunits of protein kinase A is going to go down. Okay, and in the next video what we'll do is we'll look at some pharmacological agents which interfere with this pathway.